We sometimes talk about inherited traits, as if they are only things like hair color or eye shape. But in recent decades, a more provocative question has crept into biology and the public imagination. Can life experiences, trauma, starvation, learned responses, be written into our bodies and passed to our children? In other words, do our genomes or our epigenomes store a kind of biological memory of what our ancestors lived through? To answer that, we need to separate two different ideas. The first is classical genetic inheritance, DNA sequence, the A's, T's, C's, and G's, that gets copied from parent to child. Changes to that code can and do get inherited, and they shape traits across generations. The second idea is epigenetic and regulatory inheritance, chemical marks and molecules that sit on top of DNA, or in the cell's molecular soup, that change how genes are read without changing the underlying sequence. These marks can, in some cases, be altered by the environment, things like chronic stress, specific dietary components, or exposure to toxins. These changes can then, theoretically, be passed down, influencing the biology of offspring. So when people talk about genetic memory, the idea that experiences can be inherited, they are often referring to this second, more malleable layer of information sitting on top of the DNA sequence. It's not about changing the genes themselves, but changing how those genes are read and expressed. In animals, there are clear examples of environmental conditions influencing offspring through epigenetic mechanisms, especially in simple organisms and lab models. For decades, plant biologists knew that environmental responses like vernalization, how a plant remembers a winter-to-time flowering, can persist across generations. This memory is often encoded by modifications to the proteins that package DNA, ensuring the plant blooms at the right time. In animals, powerful experiments in worms, flies, and rodents have shown that exposure to certain stresses like starvation or unusual temperatures can change small RNA profiles or chemical marks in germ cells, and that these changes sometimes alter the physiology and even the behavior of the next generation. Some of the most striking lab studies ask whether a specific stimulus experienced by parents changes measurable behavior or physiology in their descendants. For example, researchers trained male mice to fear the smell of acetophenone, a chemical that smells like cherry blossoms, by pairing it with a mild shock. Remarkably, their children and even grandchildren showed a heightened startle response to that specific scent, despite never having been exposed to it themselves. They even had more neurons in their noses dedicated to detecting it. Mechanistically, these effects are often linked to changes in small RNAs, DNA methylation patterns, or chromatin modifications in germ cells, sperm, or eggs. These act as molecular messengers or switches, carrying information across the generational divide. Crucially, these changes can persist for at least one or a few generations before often being reset. This is where things become both exciting and complicated. In controlled lab settings, where one variable can be manipulated in genetically homogeneous animals, these intergenerational effects are often reproducible. But real-world organisms, and especially humans, are messy. Populations are genetically diverse, exposures are complex and overlapping, and many confounding factors can mimic inheritance. For example, parental behavior and socioeconomic factors profoundly shape offspring environments. A parent who experienced famine might pass on thrifty eating habits to their child through teaching, not through their sperm or eggs. These social pathways create strong familial patterns, but they are not biological inheritance in the molecular sense, making it a huge challenge to disentangle them. Let's be concrete. When scientists talk about transgenerational epigenetic inheritance, they often distinguish between intergenerational and transgenerational. Intergenerational effects are changes seen in the immediate children of exposed parents. For example, a mother's nutrition during pregnancy directly shapes the developing fetus, and even the fetus's germ cells. That's technically not inheritance across multiple generations in the same way a DNA mutation is. Transgenerational inheritance, by stricter definition, refers to effects that persist into grandchildren or great-grandchildren in the absence of direct exposure. Some human epidemiological studies suggest long-lasting effects of extreme events like famines on the health of descendants. These findings are intriguing because they hint that environments can influence biological patterns across generations. But epidemiology is correlational. Teasing apart direct biological inheritance from cultural, socioeconomic, or behavioral transmission is challenging. Moreover, many early signals reported in different populations have been difficult to reproduce consistently when new data or methods are applied. So where does the field stand mechanistically? Molecular biology has shown plausible carriers of epigenetic information, DNA methylation, histone modifications, and small non-coding RNAs. Germ cells do carry RNAs and a chromatin state that can reflect parental environments. In certain animals, small RNAs appear to shuttle environmental information from soma to germline and influence gene regulation in offspring. This collection of chemical tags like DNA methylation and histone modifications gives us a mechanistic scaffold. These molecules attach to our DNA and could plausibly carry memories of an organism's experiences, acting like molecular footnotes on the genetic text. But a plausible mechanism is not proof. The challenge is demonstrating that persistent, meaningful memories of complex real-world experiences, not just simple lab-induced stresses, are reliably stored and transmitted across many generations in natural conditions. The evidence for this, especially in humans, is currently very limited. Another key point is stability. The DNA sequence with its robust double helix is an incredibly stable, long-term information store. In contrast, epigenetic marks are designed to be dynamic. Their purpose is to allow cells to adapt, so they change across cell types and over a lifetime in response to the environment. Crucially, powerful molecular systems actively reset most of these marks. This happens during the formation of sperm and eggs, and again in the early embryo. This widespread reprogramming is essential for normal development, ensuring each generation starts with a fresh developmental blueprint. Therefore, for an epigenetic change to be inherited, it faces a double challenge. It must be written into the germ cells, and it must somehow escape these powerful waves of reprogramming. While some marks are preserved, this appears to be the exception, not the rule. Understanding how often this escape happens, and under what conditions, remains a major active question. Beyond the biology, the ethical and social implications are profound. If it were proven that specific traumas could reliably pass down biologically, it would fundamentally reshape our ideas about responsibility, resilience, and public health. It might strengthen arguments to prioritize interventions for populations exposed to toxic stress or famine. But we must be careful. Overstating preliminary findings risks creating deterministic narratives about human destiny. Sound science requires cautious interpretation, rigorous peer review, and strict replication before claims influence policy or public understanding. So what should viewers take away? First, the core principle is proven. Environmental experiences like diet or stress can alter molecules in parents that influence their offspring. This is seen clearly and repeatedly in controlled lab studies on model organisms like worms, flies, and mice, where we can isolate variables. Some of these effects are robust in the lab, revealing fascinating mechanisms beyond just the DNA sequence. We're talking about small RNA molecules that act as messengers, and chemical tags on DNA packaging proteins that carry an environmental memory, subtly tweaking how genes are read in the next generation. Second, when it comes to humans, the evidence is suggestive but far from conclusive. The primary challenge is that we can't run controlled experiments on people. Human lives are complex, making it incredibly difficult to separate the subtle effects of inherited biology from the powerful influences of shared environment, culture, and social learning. Third, the field is rapidly advancing. Improved sequencing gives us a more detailed view of the epigenome than ever before. Crucially, new tools like single-cell epigenomics let us analyze individual sperm and egg cells to see what's actually being transmitted. These powerful methods will help us clarify which signals represent true, durable inheritance and which are just statistical noise. For scientists, the next steps are clear. We need rigorous multi-generation experiments with proper controls. Cross-lab replication is essential to build a reliable body of evidence, and molecular tracing, tagging, and following specific marks is crucial to confirm they are the cause of an effect, not just a correlation. In humans, integrating epidemiology with molecular readouts and, where ethical family-based sampling can help separate biological inheritance from social transmission. Finally, focusing on mechanisms is key. During early development, most of the parent's epigenetic marks are erased in a process called reprogramming, which wipes the slate clean. The central mystery is identifying which marks can survive this reset, why they are spared, and how they influence development. Answering this will turn suggestive patterns into concrete, testable models. This topic sits at a beautiful intersection: molecular biology, evolutionary theory, psychology, and public health. It invites us to rethink inheritance beyond the double helix, without abandoning the rigorous standards that make science reliable. For now, the phrase genetic memory is a useful, provocative shorthand, but we have to be careful. It risks oversimplifying a messy and subtle molecular reality. These aren't conscious memories, they are faint biochemical echoes that might slightly bias development, not predetermine it. The real story is more subtle, and, to me, more interesting. Life has multiple ways of storing information, and some of those ways may bridge generations, but we must carefully map where and how that happens. If you enjoyed this exploration and want more careful, evidence-first breakdowns of hot science topics, subscribe for weekly deep dives that separate sensational headlines from what the experiments actually show.